So, hello everyone. Welcome to today's webinar on the results of the Premier project, which is presented by people from the Premier project, which I will soon be welcoming. So today we will hear the results of an IMI project about prioritization and risk evaluation of medicines in the environment. And today's presenters will be Irene Bramke, Senior Environmental Risk Assessor at AstraZeneca in Den Haag in the Netherlands, Jan Wels, Senior Principal Scientist on Ecotoxicology and Head of Ecotoxicology at Bayer in Berlin. The start will be done by Ad Ragas, full professor in human and ecological risk assessment at Radboud University in Nijmegen in the Netherlands. And with that, I would like to welcome our presenters and start and hand over to Ad for the first introduction. Thank you.
Yes, thank you very much for this kind introduction. And I would like to start thanking the PHARMECO project for hosting this webinar. We really very much appreciate it that they allow us to share these results of the Premier project. So Premier is a project, a 6-year project funded by the Innovative Medicines Initiative. So it's now called the Innovative Health Initiative. And I hope you understand that in a 1-hour webinar, it's not possible to present all results extensively. So we mainly focus on the highlights. So I will introduce you to the setup of the project, what we aimed for in the beginning when we started the project. And then Jan Wels will take over to highlight one of our main products. So that's the Premier database and digital assessment system. Then I will come back to highlight some of the other tools and instruments and insights we generated in Premier. And finally, then Irene will conclude to tell something about how these tools and insights can be used to green pharmaceuticals, to make pharmaceuticals greener. So what's Premier about? And if you don't know nowadays how to present something, you simply ask AI. So this is actually what I did. And this is the picture that artificial intelligence produced when I gave them some of the keywords from the Premier project. And actually it quite nicely captures what we did. If you see in the top, the process, how pharmaceuticals reach our surface water and the environment. On the left side, you see some of these pharmaceuticals. On the right, in the middle, some of the organisms affected that we explore in our study in our laboratories. And on the downside, some or below in the picture, some of the solutions. So what actually was in wording the challenge that we try to address in Premier? And that's if people ask me, I've already been working about 10 years with pharmaceuticals in the environment, and people sometimes ask me, how big exactly is this problem of pharmaceuticals in the environment? I still sometimes struggle to exactly explain that. And that's basically, I think, because the environmental risks of pharmaceuticals are still relatively poorly characterized. I think the data that we need to assess these risks are fragmented, often difficult to access, for example, in industry archives. Since 2006 in Europe, we have the Environmental Risk Assessment Guideline from the European Medicines Agency, but the chemicals that were already on the market before 2006, and these are approximately more than 1,000 active pharmaceutical ingredients, largely lack environmental risk assessment data. And if we would to have gathered and test data on all these legacy pharmaceuticals, that would simply be unfeasible because this would cost an enormous amount of money. There is only limited testing capacity, and it's also undesirable from an animal welfare perspective, so all the fish testing. So basically what Premier said is we need clever methods and intelligence procedures to prioritize, to test, and to assess the environmental risk of our active pharmaceutical ingredients. So basically, the main aim of Premier is to improve, to contribute to confident, high quality and transparent data-led decision-making around APIs in the environment. Who is Premier? We are a consortium of about 25 different partners, spread its combination of pharmaceutical industry that you see on the right side of this slide, and research institutes on the left side, so like government institutes as well as universities, some SMEs, and we also had the EMA, the European Medicines Agency, as a regulatory authority involved. And as you see in this picture, we are quite evenly distributed over the different member states of the EU, and some of the pharmaceutical companies are based in the United States. But how did we achieve this? How did we contribute to actually improving decision-making on risks of pharmaceuticals in the environment? And basically, these are three components. I think on the left side, you see the data. So we generated new data on 25 case study APIs. But the most important thing is, I think we built a database on ERA data. And Jan will tell more about that in a minute. And on the other side, we developed tools and support documents for improving risk assessment. So exposure tools, effect tools, new knowledge. And on the other side, guidance documents, procedures like prioritization procedures to improve risk assessment and prioritization. And finally, the greener pharmaceuticals, which Irene will tell about. So now we're going to move towards the results. And as said, we first focus on one of the key product of the Premier project, and I will give the floor to Jan Wultz for this. Please, Jan, go ahead.
Thanks. I just took over control. Hopefully that works, and hope also you can hear me well. So what I'm talking about in the next few slides, that is to introduce to you the digital assessment system, which is the structure that hosts our project database and the tools that Ed already mentioned. What I want to introduce to you is our database. And I will already click to the next slide, and why that is so interesting and novel, and in our view is a big step forward. Well, Ed already mentioned data is partly there, it is scattered, difficult to access, and our database really helps to overcome that situation. We are building a first solution that compiles all that information, at least that information that is accessible to the project partners. And hopefully then this will be expanded later on. So it's the first central API database we have, and it provides data at a very high granularity, which allows stakeholders, for example, to assess it for relevance or reliability or for whatever purpose they need it. Then it has some key benefits coming along. It is not only a database, but it is publicly accessible. So that is one of the key problems we currently have. It is unknown what data exists, and well, you cannot often access it. We overcome that. Then it is free of charge, and we considered key user requirements, which we learned from a survey that was done earlier in the project. Good. Next question is then how to access this database. And that's very easy. You can, it's a web-based tool, so you can access it when clicking on, or better to say, typing in your browser premry.marionecri.it. And Marionecri is, by the way, the project partner that built this digital assessment system and database, and that hosts it currently. So you can use either that address, or you can also use the QR code if you like. Maybe you want to follow the next slides directly. And when you then access this webpage, then you will first see a legal disclaimer, which provides some key information. What is that information? Well, first of all, the database is still under construction. We are continuously adding new data. Then second thing is, of course, that is a legal thing. You use the data, of course, at own risk. And then one which may be more relevant to some users, and that is, while we encourage all the stakeholders to make use of the data for various purposes, and there is no limitation, it is not permitted to use the data which you can see in the database for commercial purposes. So if there are stakeholders that want to use the data for commercial purposes, they have to reach out to the data owners and to request a letter of access. And by the way, this is also the procedure that is foreseen according to the pertinent ERA guideline of the European Medicines Agency. So if you agree with all of that, then you click accept, and then you will have access to the database. And this is what you see when you accept it. You're on the landing page then, and that provides several pieces of information. And I want to guide you through some key aspects. So on the next slides, you will often see numbers, and I think that's self-explaining. We always start with number 1 and then move to the others. So what you can see here on the right upper side is number 1, and it's next to a question mark symbol or icon. And this is displayed on the landing page and provides some information, but maybe for you most relevant is there is the contact information. If you want to reach out to us for whatever reason, you can do that using this mail address, premier.das@marionegri.it. Second is, this landing page provides information on the totality of pharmaceuticals for which it contains data. And that currently is a total of about 470. I think it increased already since I made that screenshot. So, but roughly 470 APIs are already in it. And they come along with about 5,300 data records, which I think is already quite a good number. Now, a stakeholder may be interested to see which substances, which pharmaceuticals are actually listed, because we are interested in information related to that. And that information can then be taken from number 3, because there's a link to a list with information, some high-level information on the substances we have included. So you will see the compound names, you will get the CAS number, SMILES code, molecular weight, and you will also get information if the data presented has been generated with a salt or with the acid or base form, which is very important for substances that can dissociate. And that is also some very important improvement we have in our database, because typically you don't see that information coming along with the numbers of studies. But it is very important for the assessments. Good. Then number 4. Another information then on the landing page that informs you, because there may be different reasons why you access the database. But you're searching for information, and that is number 4. But you can see there is a symbol for data and one for chemicals. Data means you may be interested in all algae toxicity data. So this is the way you search for it. The chemicals path would mean you search for specific substances and want to understand which data is there for that specific substance. And I will walk us through exemplary both options. So the first option would be you're searching for data. So not for a specific data for a specific substance, but in general. And then you have 2 options. You can either populate number 1, a free text field, and for example, type in algae and then search for the information, or you can select the information from pick lists and then select if maybe you only want to see experimental data, which would be study data, ecotox, aquatic toxicity, and ending with freshwater algae toxicity data. This could be a way to search for the data you're interested in. Having done that, you will see, first of all, a high-level summary of information. And that informs you, number one, in these columns on algae toxicity, data has been found for specific pharmaceuticals. Then what study type is there? So, for example, giving information what test method has been used. And then on the right side, you also see the endpoints and the results for them. In the lower right side of this slide, you see that there's also an information on the total number of studies that have been found. In that case, 250. And then maybe you want to save this information already, and that can be done either as an Excel file or as a text file. Number three, now that you have this high-level summary, stakeholders may want to get more information, of course, on each of these studies. And that will be provided when you click here on these magnifier symbols. That will then lead you to an overview that provides this high granularity data that I mentioned. But before I show you this detailed information, I would first like to introduce other paths to search for data, because they all end with this high granularity data information. Now, search for substances. That is the second option how you search. You can search for unspecific data, all algae toxicity data, or you are interested in a specific substance and the data that comes along with that. If you want that latter one, then you're searching under the chemical path, and then there are again different options. You can type in the CAS number if you have it, or you type in the common substance name or the CAS name, UPAC name that identifies like that are all possible. And then you can search for the information. But there's also a second option that may be what you're interested in, and that is that you search for a SMILES code, or that you don't know substance name, CAS numbers, and similar, and you also don't have a SMILES code, but you have a structure. Then you can draw the molecule, because we are having a tool that allows you to do exactly this. Also, you may be interested not only in information of a pharmaceutical, but maybe of a structural element of a pharmaceutical. And you want to understand, are there other pharmaceuticals which also have this structural element? Because then maybe the information that is available for these can be useful for your purpose, maybe for a read-across approach. Then you can also draw these elements and start your search from there. If you do that, you will first see, again, a high-level summary. And in this case, it is just listing one substance, because that was specifically searched. But you could also see, if you, for example, search for a structural element, a list of several pharmaceuticals that all contain this structural element. And then again, you want to have more detailed information, and you click on the magnifier symbol. That will guide you to this overview, which provides already some more detailed information. And number one, again, informs then on what test method, data for which test method is available. It provides information on the species, what reliability assessment is there or is not there, and also what endpoints are reported with the results. Here I would specifically like to talk a bit more about the reliability assessment, because that is a very important information if you want to use the data. It is important to understand, has the data already been reliability assessed, and who did this assessment? And in this example here, you can see it has been reliability assessed, and it has been concluded to be a fully reliable study. And the term here is then always reliable without restriction. Then you can see information who did this. And it says self-assessed. Self-assessed in our database means self-assessed by the data owner. But you see a second information coming along, and that is evaluation in EU regulatory framework. That means this study has also been assessed by a regulatory body in a marketing authorization procedure, for example. So for your purpose, you can then decide if that reliability assessment is acceptable, or if you would like maybe to run an own assessment. But for us, it was important to make this information always transparent. And this is provided, by the way, for all data that we have in our database. So it's not just shown if you select this specific path to search for the data. Then on the left side, you see again number 3, and you see the magnifier symbols. And if you click there, then you will be guided to the detailed high granularity overview. Before that, however, I would like to present how the result looks if you're searching for structural elements, for example. Then you'll see this overview. Number 1 is then different. It shows you, in that case, I searched for a specific pharmaceutical that is then shown, and then the CAS number, the name of that. And on the right side, you get information on the similarity of substances listed here, similarity to your search element. So in that case, that pharmaceutical. And you see the similarity, of course, for the element itself, for pharmaceutical itself is 1. And that similarity range is always 0 to 1. So 1 means it's completely identical, and 0 means it's not identical at all. And you can see for that example here, similarity for that second pharmaceutical that is shown is 0.75. So that may help you for your purpose. And then again, you want to have detailed information on the available data, and click here number 2. Oh, and by the way, it's number 3, not number 2. You click here the magnifier symbols. Now, finally, you see the detailed information that we are providing, and I will not go in details. It should just inform you and give you an idea how high the granularity of the information presented to you is. And basically, that is all the things that you typically want to know and could extract from a study report that would allow you to understand, for example, if the data is relevant and reliable for your purpose. Then number 2 here on this slide says again that you can save that information either as an Excel or as a PDF in that case, and use it for whatever purpose you have. So that was a walkthrough on some of the key aspects of our database. And I then also am presenting very shortly information on one of the tools we have in our digital assessment system, and that is the ERA tool. ERA is the acronym for Environmental Risk Assessment. And what we are actually aiming to provide is a tool that supports key stakeholders in running these environmental risk assessments, and key stakeholders, according to our understanding and to surveys we did in this project, are, for example, water managers, risk assessors, that could be researchers, industry colleagues. So it can be quite broad. This tool will allow stakeholders to calculate environmental risk assessment according to the pertinent EMA-ERA guideline. So you have to populate some fields, but the calculations will then be done automatically, and you will get the results. And we build this ERA tool in a modular way, because stakeholders may not always want to run a full environmental risk assessment, but maybe only to do parts of it. And that may be a PBT assessment or a freshwater compartment assessment. And this will be possible because of the modular structure. And then just as a last comment, the information, this tool is not yet available on the web page. It is in a preparation phase and will then hopefully be provided in the nearer future. So that's been the bits I wanted to present, and I'm handing over back to Ed.
You're on mute, Ed.
Thank you, Irene. Very attentive. Thanks, Jan, for a nice explanation of the database. So I will quickly take you through some of the other tools that we developed in Premier, because as I said, it's a 6-year project and quite a lot came out of that. It will be a quick overview, so bear with me. We'll have a lot of detailed information. So I'll start with the exposure tools, and one of the important exposure processes is biodegradation, which is quite difficult actually to get a grip on, and also quite costly to test. Like an OECD 308 test, for example, can take up to 100 days to determine the biodegradability in sediments. So we looked, can we speed this up in a way, or can we simplify this? And one of the things we came up to it is this work from EOAC in Switzerland, is a quick screening test of 3 days. And actually, if you perform this test of 3 days, actually based on the test, you can make an estimate of what could come out of an OECD 308 test. And the prediction power, as you see on the right side, where we plotted the predicted versus the measured value in a 308 test, is quite good. So this could save in principle a lot of costs in 308 testing. Another process we looked at is sorption to soil, sorption to sludge. This is work of the University of York. They measured a lot of pharmaceuticals for sorption to sludge and soil. And for example, they developed also a QSAR tool to predict soil sorption. And then in the next step, they applied this QSAR tool to predict the vulnerability of groundwater. So pharmaceuticals actually to leach to groundwater. And as you can see on the right bottom of this slide, we can actually produce maps of areas, hotspots, risk areas for pharmaceuticals leaching to groundwater based on these QSARs. Then we made an important tool. As you might know, fish is an important target species for pharmaceuticals, since fish is, let's say, the organism that is most similar to humans, or most close to humans. And we looked at the behavior of pharmaceuticals in the fish, in order to improve risk prediction. So we developed a fish PBK model. This is non-species specific, so you can apply it to large fish as well as small fish, and it aims to predict the concentration of the pharmaceutical reaching different target organs, so depending on, of course, the pharmaceutical of interest. We compared this with the fish plasma model, which I come back to later in my presentation. And we based on also validation data, we showed that this PBK model generally is a bit better than using a simple tool like the fish plasma model. We then also looked at secondary poisoning. This was more an explorative modeling study, which still is not published, but we looked at try to model, let's say, the transfer, trophic transfer between different species levels in an aquatic system and a terrestrial system. And we modeled this as in each level as a kind of a dynamic box with different uptake and exposure routes and different elimination routes. And we compared this with validation data. Recently, in the top right of this slide, you see a recent publication where a lot of experimental data on trophic transfer were published. So we compared our model with the data from this publication. And I would say the model is doing reasonable, but not yet good enough, I would say, really for regulatory implications. But we really did make some step forward, for example, by accounting for ionization of these substances in the transfer to food webs. Then finally, the last exposure tool is, I think, one of the key products of the Premier projects next to the database. This is EPI, which stands for Exposure to Pharmaceuticals in the Environment. And basically what EPI does is, if you know the consumption of a pharmaceutical in a country and you know the per capita, so the per person consumption, it can predict based on, you should also know the excretion by that person. You can predict how much reaches ultimately the sewer system and the wastewater treatment plant. Then if you know the removal in the wastewater treatment plant, you can also predict that with, for example, simple treat. It can calculate the discharge into the surface water, and based on the hydrological system, the dilution and other processes, you can actually predict the concentration in the surface water. So we developed EPI with all wastewater treatment plants in Europe. There's a map underlying that, and with all rivers in Europe. So basically, this tool can predict anywhere in Europe the expected concentration of a pharmaceutical in a river and lake, if you know the consumption rate. And it has a very nice interface with maps where you can zoom up till 1 square kilometer resolution. And it's, I think, a very useful tool for water managers. Then I would like to show some of the effect tools we developed. First of all, we developed, this was Institute Mario Negri again, some tools to predict toxicity, especially acute and chronic toxicity for fish, and acute toxicity for earthworms of pharmaceuticals. And they did that based on read across of between compounds and structural alerts of pharmaceuticals. And these tools are incorporated in the Vera website that is hosted by Mario Negri, and you can find the code here. Then the third key product I would like to present of Premier is EcoDrug Plus. And this is a kind of a relational database that links other databases about the conservation of drug targets in different species. And so if you have a drug, often you also know what is the target of the drug in the human. And basically what this database allows you to look up that target if you know the drug. And then the database basically looks, is this target in the humans conserved in other species? Because if it is likely that that species will be sensitive to the drug. So this is mainly currently for scientific purposes, but I could foresee that in the future such a database could, for example, be used to identify species that likely are sensitive and therefore should be tested in an ecotoxicity test. We also did some work on an in vitro experiments and in vitro test. For example, in this case, you see here test to determine the absorption of pharmaceuticals in gills by fish. As said, fish is an important target species for pharmaceuticals in the non-target species for pharmaceuticals in the environment. And here we try to develop a test system where you can do an in vitro test for determining this absorption rate in the gills. It's not yet 100% stable, so there's still some improvements to be done, but we really made this step forward in vitro testing of absorption in fish. The same basically holds for metabolization. So we also looked into this. We looked at different in vitro systems. So the basic system is the liver S9 fraction that's typically used to determine metabolization. But we also looked, for example, in 3D spheroids to see if that works and is better. In general, our conclusion here is that the S9 liver fraction performs relatively okay, and that there are still some problems with developing, let's say, the spheroids and to have them stable to get stable results. So for most basic tests, the liver S9 fraction will do. Then a few words on the fish plasma model, as I said. So this is a tool that was already developed long ago, which basically reasons as follow. If you have a medicine, then typically you would take that medicine that would result in a human therapeutic concentration. And basically what the fish plasma model says, that if you use that pharmaceutical and some of it ends up in the water, in the aquatic environment, it will be taken up by a fish, resulting in a plasma concentration in the fish. You can calculate that using a KOW or something, and then you can compare, right? As soon as the fish plasma concentration becomes higher than the human therapeutic concentration, we would adverse effects to be happening. And some of the studies we did looked into this fish plasma model. For example, one study looked at, is the actually plasma binding of pharmaceuticals similar in humans and in fish? And in the top part of this result on this slide, you see that we found for most pharmaceuticals that works, but for anionic compounds, we found actually they tend to bind less to fish plasma than to human plasma, which basically means if you apply the fish plasma model for these compounds, you could underestimate the free concentration in the fish blood with a factor of 10. In the downside, you see on the bottom of this slide, you see an experiment where we try to check, okay, if this fish plasma model works, you would expect that at the concentration of a NOAC or LOAC, you would expect the concentration in the fish plasma to match the human therapeutic concentration more or less, and we explored this, and this worked for quetiapine quite well, but for two other drugs that we tested, it didn't work. So also indication that the fish plasma model works for some, but not necessarily for all pharmaceutical. Last slide on effect assessment. Looks quite complicated, but the story is actually quite simple. One of the hypothesis is, or usually when we do ecotoxicity tests for pharmaceuticals, we look at standard endpoints like survival or growth. But we know that some of these pharmaceuticals have a specific mode of action, for example, are neurotoxic, like quetiapine here. So they could affect more subtle, trigger more subtle effects like behavioral effects. And actually we could show for quetiapine that's actually true, that already behavioral effects are already happening about a factor of 10 below the NOAC for growth and survival. So subtle effects can be quite important for pharmaceuticals. Finally, I have a few slides on, let's say, how we supported the practical environmental risk assessment for pharmaceuticals. So we developed some guidance documents which explain how to perform a risk assessment, particularly targeted at water managers and people active in healthcare. So if you're interested in that and want to support that, they're not yet on the website, but they will be by the end of August. We are finalizing these documents, not only to perform exposure assessment and effect assessment, but for example, also to account for mode of action and for how to deal with data poor substances. Then as a kind of an offspin of PRAMEA, it's not something we promised, but we developed a procedure to support people to determine whether a fish toxicity test is really useful to do when you want to determine the risk of a pharmaceutical. And there the reasoning is quite simple. If you have a toxicity result test with algae and with a DAFNIT, and you know what the target is in humans, we can basically predict that whether the fish test is still useful to conduct or whether it's highly likely that the fish is less sensitive than the Daphnia and the algae. So we applied this to the 25 case study status APIs that we tested in the project, and it was shown in about 1/3 of the cases it wasn't really necessary to test a fish test because it was highly unlikely that it would be driving the risk assessment. And in this way, if you would apply this fish decision tree, you could avoid a lot of the unnecessary fish testing, saving a lot of money, but also life of fish. Finally, we also developed a lot of prioritization approaches in Premier. We apply them in two ways. So first of all, the beginning of the project, we applied these prioritization approaches to select from all more than 1,000 legacy APIs that there are on the market, 25 case study APIs that we actually tested and are performing a full risk assessment for. And we hope, we are currently optimizing this prioritization approach that has just been published, that this prioritization approach will also be applied by EMA to ultimately determine where to start testing which legacy APIs to do a full risk assessment for and which ones not. Then my last slide, and then I'll hand over to Irene. We currently also working on developing learning materials to help people and support them to learn how an environmental risk assessment should be performed and how to interpret them for academic students, for doctors and for pharmacists that are interested in this field, but also to make patients and consumers aware. And with that, I would like to hand over to Irene.
Brad?
Yes.
Can you see the next slide? Am I able to move it? Yes.
Yes, you are.
That's very good to know. Hello, everybody. Really great to be here today. So the mission statement of the Premier project, the second key objective we had was to develop some guiding principles on how one might go about doing greener drug design. And the thinking in the project, when that started, it became apparent that greener medicines actually means different things to different people. So the consortium came together and developed what we now term the greener concept. And it's an acronym where the G starts with good practice for patients, meaning that in all considerations for greener design, we must never forget that a medicine needs to be efficacious and safe in patients and is there to treat a disease. The second letter is R and stands for reduced target effects. And this is about developing medicinal compounds that are of very high specificity for the human target alone, meaning that there's a lower likelihood of existing off-target effects in patients as well as in the environmental species. Then we have two E's. One is exposure reduction, so lower emissions to start with through means such as personalized medicines or target delivery mechanisms, and a reduction of exposure after patient use through environmental biodegradability. The N in greener stands for no PBTs. So there are substances that are of particularly high concern because they combine hazardous properties such as persistence, bioaccumulation, and toxicity. And it was agreed that it is the mark of a greener pharmaceutical not to be a PBT, for instance. Then we have another E, which stands for effects reduction, undesirable moieties. So there was a recognition that there are some parts of molecules, such as perfluorinated moieties, that are recalcitrant in the environment, so very resistant to degradation, and they should therefore be avoided wherever possible. The last R stands for risk mitigation. At this point, where a medicine is in existence, there should be methods developed to mitigate any environmental risks that it might nonetheless be posing. So that was the starting point. And then we moved on to the first deliverable under Premier. And this was a report. So I'm always including the QR code and a link to the actual Premier report, and then where a publication resulted from that as well on the slides for anyone who has interest to learn a bit further. But here we investigated how the drug discovery process actually works and where the right points are to intervene if you want to end up with a greener product. What the key attributes are that predictive tools and assays must possess to allow medicinal chemists to use them alongside their current ones. And the key outcomes really of this work here was that we got an understanding that intervention during the R&D process really needs to happen very early, so long before one is settled on a development candidate. And that is because the molecular properties are actually set at the design of the API. And then we had the second deliverable, which was a report summarizing the fundamentals of drug design. And here we dived really deeply into the science and tried to tease out what it means to be designing a medicine with only the patient in mind. And here the patient is conveniently depicted as a gray circle. So currently, active pharmaceutical ingredients are designed to be potent, to be specific. We are designing them to use structural modifications to design for certain physicochemical properties. And at the same time, you're also striving for abiotic stability to ensure shelf life and reduce the risk of the formation of reactive breakdown products under storage. And this is achieved through many iterations of the so-called DSTA cycle, so design, synthesis, test, and analyze. The projects are successively undergoing and creating increasingly optimized analogs by doing that. But at the same time, and this is exactly what medicinal chemists aren't trained to be thinking about, is, for instance, if you are designing an API to have a reasonable lipophilicity because perhaps it's a neuroactive compound and needs to go through the blood-brain barrier, you might also inadvertently be designing something that is bioaccumulative in wild species because the breakdown mechanisms that prevent bioaccumulation in the patient aren't the same as in wild species. At the other end of the lipophilicity range, you might be designing a compound that has significant mobility in the environment, leaches into groundwater, and causes a problem then in drinking water production. Also, by designing the compounds to have sufficient metabolic stability within the patient so that you achieve an effective internal concentration and systemic exposure, you might at the same time be closing down routes for breakdown in the environment, and you might inadvertently be designing this molecule to be environmentally persistent. And then last but not least, by designing excellent potency against a human target where a homologous target exists in an environmental species, you are then inadvertently designing something to be ecotoxicologically potent. And then the dose determines the environmental input, and you combine that with the persistence and mobility of the compound, which gives you an understanding of the likely level of environmental exposure, which is then compared to the ecotoxicological effects concentration, which gives you an understanding of the environmental risk. So this is all very interesting and very helpful, but it is cumbersome and not very easy to explain. So with the next deliverable, we then actually moved forward towards making proposals for implementation. So we developed this simplified conceptual model of the environmental impact potential of APIs. But in the light yellow boxes here, you will recognize many of the criteria that I've just discussed. So the size of the administered dose, the extent of excretion, the number of patients that are being treated with a certain drug, all inform the key driver that we call input. The things that determine the second key driver, which we call output, that's the potential for the substance that is made and used by patients to then be reduced after use through breakdown in the environment. And then thirdly, ecotoxicity, which determines ultimately the risk. Again, there's a Premier report with more detail for anyone who has interest in reading further. But we've then simplified that down even more to something we call the cube. So this allowed us then in the conversation about this cube to recognize that not all R&D projects are created equal. Some pharmaceutical R&D projects aim at targets where there is a very low likelihood of a homologous target existing in a wild species. They might be addressing a rare disease with very few patients to treat, or they might actually be dealing in an area of chemistry where there's a reasonable scope for environmental degradation. So they are perhaps not the area of prioritization, but at the other end, there are then R&D projects that live in the right-hand side corner of this, and those should be prioritized. For attention, for intervention in terms of green design. So we are shaping this into a guidance document, which we're going to use to publicize and popularize this concept of the green operating space and advocate its use in practice. But then there are other things that also need to happen. There's still more work to do. There needs to be greater exchange between environmental experts and medicinal chemistry R&D experts. We still need to work towards agreeing harmonized criteria and thresholds. This isn't something that an environmental expert can do on their own or a medicinal chemist can do on their own. So this needs to happen in cross-functional teams. And of course, we need predictive tools and assays that are actually able to fit into the modern R&D process. The DSCA cycles that I've already mentioned, that's tens to hundreds of compounds tested within a cycle of a week or two. So any assay that's to be used to provide information for decision-making on environmental grounds needs to harmonize with that and needs to work on the same clockwork cycle. And as far as predictive tools are concerned, medicinal chemists need to be able to trust those tools. So it falls to the environmental experts to explain what it is that a prediction tells us, where the uncertainties are. So there is still a challenge there for us in the future. But we also need to look beyond the technical, because it is more than a technical challenge that we're facing here. It's also important to realize that the cross-functional teams that are doing R&D in pharmaceutical companies don't currently include environmental experts. I've already mentioned the necessity to bring that closer together. But the R&D projects actually have other stakeholders in them. So there's overall project leads, for instance, that are potentially not chemists and are definitely not environmental experts. But these individuals, these portfolio leads in pharmaceutical companies, they hold the responsibility for the entire pipeline, and they are not yet sufficiently aware of the environmental challenges that arise from the APIs that are being designed in the projects that they're looking after. So this conversation needs to involve the experts in the regulatory functions, the business leads, because at the end of the day, pharmaceutical companies are businesses. And really only after this has happened, and it can be done, it's happened in the pesticide industry 10, 15 years ago, so this isn't an insurmountable challenge, but it is a challenge that we are at the beginning of in the pharmaceutical sector. And only once we've gone through that and had those conversations and had that opportunity to educate the wider experts involved, will we really understand the true operational feasibility of doing greener design in earnest and what the scope is to have beneficial outcomes for both patients and the environment. And that was my last slide. Thank you.
Thank you very much, Irene, Jan, and Ad, for a great presentation today. With that, I would like to open the round of questions. So there were quite a few questions in the chat, part of which were already addressed by the speakers today. So I would like to just raise the questions that were not yet answered. So first question is about, were there any exclusion criteria applied to the data being submitted to Premier?
Yeah, and unless you want to take that.
Yeah. Well, basically, the commitment was that all the companies, the FPA partners in that project, provide the ERA data which they have. So that is that used in marketing authorization, and that's typically also any data which exists beyond marketing authorization. But the question was if there have been any exclusion criteria as such. And I think not in the database. In the prioritization exercise, we excluded, for example, specific modes of action, but not in the database.
I think in the Premier project officially it didn't focus on antibiotics and what was the others? Hormones, I think.
Yeah.
So these were not considered, but they, for example, in the database, they have been entered if there is an EPAR report on them. So, yeah.
Yeah, also project partners have provided this data, even if it was not in scope.
Yeah.
And maybe linked to that, what is the plan for maintaining, developing the Premier database now when the project will end?
I would give it a start, and the others may jump in. I said this is a question for Q&A because I wanted to make sure that's of broad interest and that everybody is seeing that answer. Now, that's something that is driving us a lot and where we don't yet have an answer, but there are different levels, let's say. First level is definitely the project partners do not want to have a project database that is dying with the end of the project. That would be really a shame, considering all the investments we made in that. So project partners are now in sustainability discussions, how we can make sure that, first of all, over the next 2 years, the database is maintained, and potentially only also that further data can be uploaded. How that will exactly look like to be decided. This is a running process. Then there are more aspects to be considered, and they are beyond the project. So, I mean, there is now this really nice central database that provides this high quality and high granularity data. And what we clearly hope that this is then a starting point, and the database can be set at a higher level and continued at a higher level. For example, we see the revision of the pharmaceuticals legislation that is probably being adopted end of this year. And there you can see a request for EMA also to build somehow a data repository. So there are several aspects that are all not answered yet. And we see there are options, and it seems to be all in a good way that this database is continued and that it can be expanded, and that maybe and hopefully it will really be a starting point for something really big. But yeah, there's not a lot more I could say now because we don't have the precise answer yet.
Thank you, Jan. I think you answered that already in the presentation. However, I would like to reiterate, did Premier generate any full ERA for any APIs?
Yeah, so we selected 25 legacy APIs to be tested by the pharmaceutical industry. So the different pharmaceutical industry partners adopted 2 or 3 APIs per partner, and they tested and ran in principle a full run of tests. So for KOW, algal toxicity, and Daphnia toxicity. And if the fish decision tree said it was useful to test the fish, that was also conducted. So for 25 APIs, we have run tests, and we will produce also a risk assessment for these 25 APIs.
Thank you. We have a question. In the Premier database, information is provided on whether a reliability control has been conducted by EU regulatory framework. Does this include other regulatory EU bodies than the medicines agencies that assess the data during the marketing or authorization procedure? If so, it would be valuable to get information on which regulatory body assessed and accepted the data.
So I'm happy to take that one. So this isn't actually always an easy question to answer, exactly which member state or who the assessor was at marketing authorization application, where you see that it's been assessed for reliability other than self-assessed. That typically means that the API in question has got an EPAR currently live, so it has a live registration currently going, and that's all the granularity that we can consistently provide.
Maybe related to that question, thanks, Irene. Are there any plans to work with EMA to include some of the prediction methods in the ERA guideline?
Well, there's always the hope that when the environmental risk assessment guidance gets revised in light of the revised general pharmaceutical legislation, when that working group reopens, that there's going to be cross-stakeholder engagement on the redesign of that environmental risk assessment guidance, but we have not heard anything to that yet.
All right. So the last question was just answered by Jan in the chat, which was EMA specified that read across and QSAR tool prediction are not accepted for ERA. How will it be addressed in this ERA tool? So you answered the ERA tool that Premier builds enables ERA coding to pertinent EMA guideline. That means QSA, R, and or other data currently cannot be used. Thank you for that answer. Any other questions right now? I don't see any further questions in the chat. So are there any other questions?
Maybe one question I saw coming by was whether the EPI tools is based on human pharmaceuticals only or veterinary medicines as well. So currently it's only using human pharmaceutical use. It cannot predict releases from veterinary use. We are working on models to improve that, so to add a veterinary component, but the EPI currently only deals with human pharmaceutical consumption.
Perfect. Thank you, Art. I missed that one. If there are no more questions, I would like to thank speakers of today's webinar. Thanks, Irene, thanks, Jan, and thanks a lot, Art, for great presentations. And thanks all in the webinar today for joining this webinar, and thanks a lot for staying with us and raising your questions, and looking forward to seeing you in one of the next webinars. Thank you very much. Bye-bye.